All right, I think we'll get started here soon. So, if you guys can hear me, just go to the attendance chat, let me know you can hear me, or just give a thumbs up if you guys can hear me coming through and if everything sounds okay. And if it's all working, then we'll get rocking and rolling. Cool, perfect. It looks like people can hear. So as we go through this, at any point, if the sound cuts out, if the presentation, if anything kind of goes wrong that I don't catch on my end, please just let me know with a direct message, and then I'll kind of stop and try to fix it. Um, so if you guys could save questions for the end, you can ask them. I probably won't get to them till the end. We only have a short kind of period of time, so I want to make sure we get through all the information, and then at the end, I can go through all your guys' questions. We can answer them and talk a little bit more. So it's 6:03, so we'll get rocking and rolling. And if people come in throughout the chat, then that will be perfect. Let me just check there's one more message real quick. Perfect. So today, the title of the presentation, Putting an End to Dementia, right? So today's topic is largely going to discuss dementia, more specifically Alzheimer's. We know more about it. There's a little more data about it. It's more common than some of the other forms of dementia. We'll talk a little bit on those, but today we're really going to talk about cognitive decline. What really goes wrong in the brain? How can we test it? And what are some things we can do about it? Okay. Yep. Sorry, the computer's being a little slow. Perfect. So, a little about me before we get started. So, I recognize some of the names on here, others I do not. So, my name is Dr. Perry Maynard. I am a diplomate of the American Board of Chiropractic Neurology. So, originally, I am a chiropractor, but have a diplomate in clinical neuroscience. So, what does that mean? Is that I did some extra schooling in clinical neuroscience, right? So, chiropractic by trade, but mostly what I do work with neurological disorders. So, I work at a clinic in Denver. It is Integrated Health Systems. And the majority of the conditions I see tend to be TBIs or traumatic brain injuries, concussions. I see a lot of vertigo and dizziness patients, cognitive decline, various types of autoimmune conditions. So things like Hashimoto's or celiac disease, MS or multiple sclerosis, things along those lines. And then things like dysautonomia or things like POTS or postural orthostatic tachycardia syndrome or even orthostatic intolerance. So see a lot of things surrounding the brain, right? And so today we're going to talk a little bit more on the topic of cognitive decline, right? So the goals for today, right? I want to discuss symptoms associated with cognitive decline, right? A lot of people say, well, I forget things all the time. Is that big of a deal, right? So we'll start to kind of outline how do we categorize people with dementia? When do you need to see a doctor? When may it be okay? We're going to discuss the metabolic processes behind Alzheimer's disease, right? So, tell you a little bit about what's going on chemically and in the brain to develop things like Alzheimer's disease. We're going to discuss different subtypes of Alzheimer's disease. And for some of you, this may be like, wait, what? There's different types, right? So we'll talk about some theories about different types of Alzheimer's disease. We'll discuss testing, right? And then we'll discuss things that you can apply today. Okay. So let's look kind of at some different things, right? So a lot of people talk about genetics, right? And I hear genetics a lot of times get thrown around kind of as the blame for everything. And although genetics plays a huge, huge role in understanding genetics and genetic markers in certain pathways is very, very important, we need to more understand how is our environment affecting our genes, right? We're seeing such a huge rise in things like Alzheimer's, right? Because of the baby boomer surge, right? There's so many more people at this time getting into their 60s, their 70s, getting older, right? So we're seeing more of these development of neurodegenerative diseases. Also, the other big thing is that I think we're seeing a lot more of it because lifespan is expanding. For the longest time, people didn't live beyond 30, 40, right? And a lot of these disorders and neurodegenerative disorders are disorders of time in aging. And so we'll discuss all the different factors. There are some things that can cause it to occur quicker and other things that can cause it to occur at later stages. So let's look at some stats, okay? Alzheimer's disease is the sixth leading cause of death in the United States, right? That's pretty crazy and baffling that it's up there as one of the top leaders of death, right? It also plays a huge role in bankrupting a lot of families and a lot of individuals. Genetic susceptibility accounts for only a small percentage of cases of cognitive decline, right? We'll talk a little bit about some genetic markers that play a role, but there's far more to the story than genes, right? Most of the cases or most of the things with neurodegenerative disorders, right, they occur from breakdown in pathways that have to do with inflammation, immune regulation, right? All things that break down with age, right? But certain environmental triggers can help to accelerate that process a little bit quicker. Right? So what are some symptoms that we hear on a daily basis, right? You hear people all the time, where did I put my keys? Where did I park my car? What was their name again? Wait, what was I gonna say? Right? These all things that we hear people say on a daily basis, right? And these kind of explain what are called the five domains of cognition, right? And this is how we kind of categorize individuals with cognitive decline to determine how far gone are they? What systems are really affected, right? So the five domains are visual spatial. Where did I put my keys? Where did I park my car? Where am I in the world? Language, difficulty with understanding language, speaking, uh, forgetting what words to say, acquiring and remembering new information. So having a hard time learning new things. Maybe you started a new job and you just can't pick up things quite as fast, or you used to play musical instruments and you just can't pick up new instruments, right? Trouble picking up and acquiring new information, right? Reasoning and judgment, right? You see this a lot in patients with Alzheimer's or kind of leading that direction, right? Where the things they start doing just don't make sense, right? I've seen this with grandparents in the past where someone calls and next thing you know, grandma is giving all of her financial information to someone she doesn't even know over the phone, right? Not using good judgment, right? And then personality and behavior changes, right? People start saying, this isn't the individual I married. I don't recognize this person. They seem different, right? So we start seeing changes in personality as well. Oh, let's go back. So as we go through this, right? So those are the five domains, right? We're going to start going through some categorizations and we're going to see how those domains, when they get affected, how they kind of go with the progression of dementia. So the first thing we see, and this is probably the most common that we see, and I think this is the most common that gets overlooked by physicians, right? And really, if you catch an individual in this stage, you can make huge improvements. And I think, I would argue that you catch individuals in this stage and you may be able to halt or keep certain things and certain neurodegenerative diseases from ever actually happening, right? And I don't have any evidence or research to say prove that, right? But these are individuals really in that early stage, and I think this is when interventions are the most fruitful, right? So this is what's called subjective cognitive impairment. These are individuals who say, my memory just doesn't seem there, right? I just don't feel like things are clicking as well. I'm depressed. I'm feeling overwhelmed. A majority of the patients I see have a majority of these symptoms, right? And they go to doctors and they say everything is normal, right? They have all this cognitive testing done, imaging testing, everything's done. So they tell them, well, it's in your head. I don't know. It's not a big deal. But who's to say these aren't the early signs, right? There's a lot of research showing that these disease processes, they start 20, 30 years before the first real symptoms occur, right? So in one's 20s, in one's 30s, right? These little occasions where you say, ooh, I feel a little foggy, if left untreated, could down the road end up into some of these things. Now, it could be nothing and it could end up there, but that's why the sooner we can get on top of these things, the better, right? So that's subjective cognitive impairment, right? The next thing we're going to discuss is what is called MCI, right? Or mild cognitive impairment. So mild cognitive impairment occurs when any of the 5 domains are significantly impaired. So remember, visual, spatial, language, personality, behavior, reason and judgment, acquiring new skills, right? Any of those become affected along with being able to maintain independence in social and occupational functions. So these individuals that are still working, they're still getting things done, but man, they really have trouble knowing where they park their car, the keys on a daily basis, right? This is where you start getting into mild cognitive impairment, right? And this is kind of a step further from subjective cognitive impairment. Still, this is a good stage that if individuals are caught and treated appropriately, a lot of good things can be done, right? And I think once again, some would argue there's a lot of great research out of UCLA by a researcher named Dale Bredesen, and a lot of this information is kind of from him and things that I've learned from him. But he's got a lot of case studies that he's starting to publish of being able to reverse and slow some of these things, especially when caught in the mild cognitive impairment realm. Right. And then we have dementia, right? So what makes it dementia, right? Dementia is when at least 2 out of the 5 domains are affected, right? So you may have language issues and personality issues. You may have visual spatial issues and you may have difficulty acquiring new information, right? And this is when it really serious, unable to work, unable to pay bills, things like that. That's when things really start to get serious. And this is where things like neuropsych testing are really important to kind of measure these different domains, right? And this is a farther progression from subjective cognitive impairment and mild cognitive impairment, okay? So let's talk a little bit about different types of dementia, right? We talked about categorizations, right, and severities, right? So some of the most common that some of us may hear about are Alzheimer's, Lewy body dementia, things like Huntington's, frontal temporal dementia, and things like multiple systems atrophy. Some of you may never have heard of any of these besides Alzheimer's. Some of you may be familiar with a lot of these, right? We're not going to get into all these. We're only going to get into Alzheimer's tonight because we could be here for weeks discussing all of these different types of dementias, right? But these are some of the more common types that there are, okay? So let's get into Alzheimer's, right? So before we get deep, I want to talk a little bit into mechanisms, okay? So I want us to think of the brain, more specifically neurons, right? So the brain is made up of neurons, right? And those neurons have what are called synapses, and these are bridges that help communication between different neurons, right? And you have receptors, right? So all these neurons have different types of receptors on their cell surface, right? And these receptors receive information, or they have certain things bind to them. So you can see here in the picture, you have a ligand, right, which could be a protein of some sort, and you have a receptor. And when they connect, they code for a certain thing to occur, right? So in certain occasions, that ligand might bind to that receptor and signal for the cell to die, or it may bind and signal for the survive. So as we go through talking with dementia, I want you to think about it is that it's a problem with signaling and signaling pathways, and that certain things are tripping a switch that is signaling for too much cell death versus cell survival, right? So almost like a downgrading or a pruning where the brain doesn't perceive that it's a very enriched environment to survive in. So it's going to down regulate things to stay alive. And this is why Alzheimer's patients, right, their basic needs for survival stay intact for a long time, right? They can breathe, their heart rate, their blood pressure, all these vital things stay intact, but the brain starts to downregulate things that it deems to be not that important, things like memory, right? Because really at the end of the day, to survive, you don't really need to know who your neighbor is or where you parked your car, right? You need to know how to breathe, you need to know how to eat, and all these different things, right? So we're going to talk about this model a ton of the receptor and the ligand, right? So on those neurons, you have that receptor, and different chemicals and compounds will bind to that receptor and will determine the fate of that neuron. It will determine whether it lives or whether it dies. So a ligand can be similar to a neurotransmitter. A lot of times, ligands are different proteins or amino acid structures, but yes, definitely. Neurotransmitters bind to receptors all the time on the end of synaptic clefts within the brain to signal certain things, right? So they're all just different, protein sequences or chemical makeup and things like that. But you can think of it in a similar way, definitely. So we're going to get a little bit of new stuff, so hang on with me for a second. I won't stay on this too long, but we're going to talk a little bit more in depth about really what occurs, right? So some of you may be familiar with what's called beta amyloid plaque, right? So amyloid beta is a protein, or it's a peptide, right? So it's a sequence of amino acids or chemical structures, and they make up a peptide, which is a signaling molecule, okay? Beta amyloid or amyloid beta is kind of like the hallmark sign of Alzheimer's, right? So it's a protein found in patients with Alzheimer's that starts destroying synaptic connections, okay? So if that's the case, we need to figure out, why does the brain start creating amyloid beta or beta amyloid plaque, right? Because if we can figure that out, then we can try to intervene to keep from increasing that protein structure, okay? So we have something called amyloid protein precursor, right? So this is, once again, this is like a little receptor. It's a structure that sits on the neuron, right? And it's just kind of examining the outside environment and deciding, is this a good environment? Is this bad environment, right? And certain things can bind to this, right? There's also certain things that can cut this as well, right? So within your body, you have certain things called certain enzymes that have to do with cutting certain fragments into certain lengths, and depending on what length that gets cut, think of it like spaghetti, and spaghetti can cut into long strands, short strands. Those code for different things, okay? When that APP, or that amyloid protein precursor, is cut into 4 segments, it creates pro Alzheimer's types of almost kind of like genetic makeup, right? Or signaling to start creating more amyloid beta or beta amyloid plaque. If you cut it into 2 segments at a different site, it creates things that actually are protective against Alzheimer's. So it does things that help with growth. So you can see we're just cutting a certain peptide at a certain point totally changes the signal your brain gets. So what things bind to create the good cut, and what things bind to create the bad cut? So we have something called nectrin, right? And we have other certain types of peptides, right, that can bind to that segment, and they create a cut that creates 2 segments, and everything's great, right? And then, funny enough, beta amyloid or amyloid beta, right, the plaque itself actually binds and creates that cut into 4 segments and causes a signal for death. So the more beta amyloid plaque you create, it becomes this vicious downhill spiral where you create more and more and more and more, right? So the question is, how do we kind of move the balance towards what I like to call synaptogenesis, the creation of new synapses and new neurons, versus synaptic activity, which is the breakdown of neurons, right? And if we cut that segment into 2, we get genesis. And if we cut that segment into 4 segments, right, we get breakdown of neurons. So we got to talk about, what are all the things that will push the dial in either direction, because those are the things that really need to look at in order to have the best outcomes with patients. Okay. So this is a paper, but also a picture. Once again, kind of a busy picture that I don't expect you guys to understand or really dive into. But this is taking a look at that secretase or that scissor, that cuts that APP or that precursor protein in those either 4 or 2 segments. You can see where non-amyloid or amyloid producing, right? So we want to shift it towards the green. We want to do things that are going to shift cutting that peptide or that protein in a beneficial manner, right? So what are those things, right? That's kind of the million, or I should say $1 billion question. Okay. So a lot of this is going to be, like I said, from the work of Dale Bredesen out of UCLA, who's a clinician and researcher, right? And most of his research has been to look at all the factors possible that create that shift, right? So all the things that will create that cleavage or that cut into 4 different segments, right? And as of currently, they've discovered 36 different things or factors, right? But I'm sure there's hundreds, if not multiple hundreds, that we'll kind of discover as the years go on, right? So he uses an analogy of 36 holes in a roof, right? So let's say your brain is that roof, right? And you have those 36 different holes or those factors that are creating that shift towards synaptoclastic activity or breakdown of neurons, right? If you plug up 1 or 2 of the holes, it's still going to flood. You plug up half of them, it's still probably going to flood, but maybe not as bad. You have to do your best job to plug up as many holes as possible to keep the building from flooding, right? We have to go after as many factors as possible to give us the best opportunity of creating synaptogenesis or growth in new neurons, right? So I love this analogy that Dr. Dale Bredesen has made of this 36 different holes, right? Because as we go through this, we're going to talk about all these different factors that can play a role in this. Okay. Right. So let's talk about this. Right. So Dr. Dale Bredesen has started to classify different subtypes. Um, so sorry, I was answering one of the questions on there. So there's 5 different types that we're going to look at. Okay. One is going to be an inflammatory subtype. One is what's called trophic. The next is called toxic, glycotoxic, and vascular, right? So these are different subtypes. So now maybe we have a person or a patient with Alzheimer's, but how do we classify them? What are the driving forces? Do they fit more into an inflammatory model that's punching holes in the roof, more of a toxic model, more of a blood sugar model? So we'll kind of talk through all these different things, how we test them, and then ultimately, how do you treat them? Okay. Shift to the next one. Okay. So if we look at type 1, okay, so type 1 are hot or inflammatory, right? So this is the first one where we're looking at individuals who tend to carry one or more of what's called the ApoE4 gene. Okay, so this is where we're going to talk a little bit about genetics, but not too much, right? So the ApoE4 gene, some of you may be familiar with, some of you may not. Some call it the Alzheimer's gene, right? But it's a gene associator seen very closely and seen with an increased risk of developing Alzheimer's, right? And it's an inflammatory gene. And this is going to be a little side tangent for a second, but I think it's kind of interesting. So we developed the ApoE4 gene, right, when we went from being tree-dwelling animals to ground-dwelling animals. And the theory is that now that we're on the ground, we have much more of a likelihood of coming across parasitic infections and all sorts of things that could create all sorts of destruction. So the goal of the ApoE4 gene is actually inflammatory, right? So it helps to fight off brain-eating parasites, brain-eating amoebas, things like that. But unfortunately, it leads to a lot of inflammation in the brain, right? And this is why individuals with 2 copies of the ApoE4 gene can be upwards of 50% increase of development of Alzheimer's, okay? But this is where we see things like increased CRP or C-reactive protein, decreased ratios of albumin to globulin, increase interleukin-6, increase in TNF alpha or tissue necrosing factor, and then things like insulin resistance and increased homocysteine. I know these are a lot of big words for some of you, or maybe even a lot of you, and we'll kind of go through them individually. Okay, so the first thing we are going to talk about is that CRP marker, right? So what is CRP? What does it stand for? Why do we care, right? So CRP is high sensitivity C-reactive protein, okay? And when this is elevated, it indicates chronic inflammation is occurring, right? So it's used in a lot in the cardiovascular world and with heart attacks, different things like that, but it's a very non-specific marker, right? And a sign of inflammation occurring, right? So it's just a sign, right? So as we're looking through blood work, we want to see how many boxes can we check where we can start putting patients or individuals into certain categories, okay? The next factor is going to be what's called homocysteine, right? So homocysteine is, oh, sorry, skipped a little far. So homocysteine is going to be a really important marker to look at because there's more and more research that's developing looking at its role in strokes, depression, autism, psychiatric issues, all sorts of things because it can be very inflammatory to blood vessels, but also the brain as well, right? So I want you to think of it almost like someone running through your blood vessels or through your brain with a knife, just kind of tearing things up, okay? Homocysteine, right? This is just a byproduct that naturally occurs within the body, right? So you have what's called methionine, and methionine is an amino acid, okay? All right, and you kind of cycle it between homocysteine, methionine, back and forth. This is a huge role in detox pathways, right? So some of you may be familiar when individuals talk about methylation defects or MTHFR defects, that can play a role in that homocysteine level as well. Okay, but there's a lot of interesting research going back to looking at that homocysteine molecule and showing atrophy in different brain regions, slowing of cognition, and things like that. And actually, when treated with certain types of methylated B vitamins that can actually lower homocysteine, actually reversal of some of these things as well, right? So homocysteine is going to be a really important marker to look at. Okay, now we have type 2, right? Type 2 is atrophic, or what they kind of call cold. Okay, so this one I want you to go back to. Remember when we talked about that ligand or that receptor? Okay, that has to tell whether it's time to grow or whether it's time to die, right? The brain and the body are always using these signaling molecules to know whether it's time to thrive or whether it's time to recluse, okay? So this is a loss of trophic factors, right? So these are the patients, right, where there's suboptimal hormones. So things like thyroid hormone, estrogen, testosterone, progesterone, pregnenolone, vitamin D, right? All of these things are trophic factors, right? So they're things that encourage growth, right? Thyroid hormone is your furnace, right? It gives you energy. Estrogen and testosterone have a ton to do with neuroprotectivity, both females and males, and they have everything to do with growth, right? And same with vitamin D, right? So I want you to think of these structures as miracle grow for your brain, right? So some of you may be familiar with something called brain-derived neurotrophic factor. Okay, this is miracle grow for your brain. This helps you to create new synapses. So thyroid hormone, estrogen, testosterone, vitamin D, all these things help you create new synapses, right? These help to bind to that ligand receptor or to that receptor to signal, stay alive, it's time to grow, this is a healthy environment. When you start getting suboptimal levels of these hormones, your brain's going to start getting messages saying, hey, this is not a great time. We need to down regulate our connections in order to save energy, right? So, this is a really, really important thing to look at. This kind of goes back to those 36 different holes, right? So, these play up, say, 10 of the different holes that need to be punched up. So, when we're looking at patients saying, hey, there's thyroid issues, they're postmenopausal. This is a big reason why you see females, especially postmenopausal females, are at a much greater risk for dementia and Alzheimer's. And you see it more often as well. And it goes around with estrogen and some of these neuroprotective compounds. Okay. Next, we're going to look at type 1.5 or glycotoxic, right? Or sweet. These are the ones where they're more associated with elevated glucose or individuals with diabetes, type 1, type 2, type 1.5, whatever it may be, right? So these are the individuals that have high insulin levels, they have insulin resistance, they have type 2 diabetes, right? All of these things are going to affect neuroinflammation in the immune system. In a second, I'll kind of show you how insulin and insulin levels play a role in creation of that beta amyloid plaque. right? So when we talk about glycation, right, what we are talking about is the ability to sugars to bind to DNA structures, protein structures, peptide structures, right? All these signaling molecules and change the conformation of them, right? So glycation is when sugar binds to these things and then sometimes renders them useless or changes the code. Remember we talked about code is everything. The signaling molecule, it has to be so specific, otherwise you can go down two different roads, right? So this is why high blood sugar, right, can start to change protein sequences in the way your brain receives messages. Okay. Oh, let's shoot to the next slide. Okay. So like we talked about, right, sugar binds to proteins and fats. Oh, and changes DNA. Okay. The big thing with insulin and insulin resistance. Okay. Can you guys still hear me? I know I just had an individual saying I was cutting in and out. Are other people having trouble hearing me as well? Okay. So let me know if. Okay. So it looks like people are good. So let me know. Okay, perfect. So going back to it, right? So discussing the high blood sugar, right? So insulin, right? A lot of people talk about insulin resistance and why it's so important, right? So when you look at right excessive sugar consumption or things like that, or even type 2 diabetes, right, you start to insulin plays a role in shuttling fuel into cells. So it takes sugar out of the bloodstream and it moves it into cells for energy, right? You can start becoming resistant to insulin to where you start signaling from even more and more insulin because you're becoming insensitive to the peptide, right? And this will create insulin sensitivity and higher levels of insulin, right? Higher levels of insulin are not a good thing, right? And they can be very inflammatory. And why this is you have something called insulin degrading enzyme, or IDE, right? This enzyme plays a role in breaking down insulin. Unfortunately, this enzyme also plays a role in breaking down beta amyloid plaque. Funny enough, right? It can only do so many things at once. And if there's more insulin present, that's less enzymatic activity that goes towards breaking down beta amyloid plaque, right? So elevated levels of insulin will decrease your ability to break down and to clear the peptide or the protein associated with Alzheimer's. Okay, so that's why insulin and insulin resistance is a really big factor with the development of Alzheimer's and is a really easy thing to mitigate, right? And to improve on patients and things like that. We won't get into that fully in this discussion, but in other lectures, we'll discuss different ways of improving insulin sensitivity. Okay. Here's a picture. You can see here normal healthy synapse, okay? High insulin levels. That IDE is used up on the insulin, so you start getting that buildup in that beta amyloid plaque. And then you can see that end result is that beta amyloid accumulation starts to create that neuronal dysfunction. Okay, so we need to drop insulin levels and improve insulin sensitivity so that enzyme can do more in regards to breaking down that plaque that's not so great for our brain. Okay. And then the last type I'm really going to talk about tonight is going to be type 3 or toxic. And this is one I think is really, really interesting. And when I learned about it, it made a lot of sense. And I don't think there's a lot of people discussing it. I think it's kind of gaining more speed, but it's the toxic form. Okay. These are the individuals where they don't really make sense. They look like multiple different forms of dementias, multiple areas are affected, can look like vascular changes, right? And this really you see with patients suffering from chronic infections, right? Or immune dysregulation, or how some would categorize Alzheimer's as an autoimmune condition, right? To where you are now creating antibodies against your brain. So this is more the toxic model is more surrounded around immune dysregulation and how it's affecting the brain. Some of the most common infections that you can see or what they're finding are things like Lyme disease, Babesia, which is a parasitic infection, and then things like Bartonella, right? These are co-infections that come with Lyme disease. And what's interesting is they're starting to do autopsies on patients with Alzheimer's and they're finding Borrelia burgdorferi, which is the bacteria, the spirochete associated with Lyme disease, right? So some individuals are starting to associate things like Lyme disease with Alzheimer's and other neurodegenerative disorders, which I think is interesting. It makes a lot of sense. And there's even some discussion that actually the beta amyloid plaque has antibacterial properties as well. So are we seeing elevated levels of beta amyloid plaque because of an infection, right? So we try to drop those levels, but really it's just a side effect, or it's just a sign of something more serious going on. And then different types of viral infections, more specifically the herpes family. So different herpes simplex viruses and HSV9 and 6 and all these different things, right? They have more of an affinity for the brain, right? And all these different infections can just create inflammation and irritation within the brain. Another big thing that's gaining a little more momentum is mycotoxin exposure or mold exposure, right? And then heavy metals and then other chemical exposures, because ultimately all of these infections and mycotoxins, they're just biotoxins, kind of like anthrax, right? So being biotoxins, they just dysregulate your immune system and they affect the way that that APP is cut, right? Or that precursor protein is cut, which is then going to shift you towards more of a breakdown mode versus a growth, right? Because if you have a chronic infection, your body and your brain's going to say, this is not time to replicate. This is not time to form new memories. We got to survive. We got to get rid of this infection, right? So think of it almost like as an adaptive scenario, which I think is kind of an interesting theory or way to look at dementia. Okay, so I love this term, and this term was coined by, once again, kind of the doctor I was talking about earlier and some of the other different researchers, Dale Bredesen. And it's what's called cognoscopy. And I love this, right? Because everyone talks about getting a colonoscopy and, when you're getting into your 50s and getting all these things checked. But what about a cognoscopy? No one talks about getting checked out for your mental health, right? When I say mental health, I just don't mean mental well-being. I mean, how well is your brain working? How are things affecting your brain, right? Especially you start getting to that 40s range, that's when some of these processes are really occurring, right? So cognoscopy is something that I would recommend for anyone above 45. And honestly, anyone in their 20s and 30s, I don't think it would hurt to start looking at these things to try to make these numbers optimal, right? And the cognoscopy, right, it will include comprehensive lab work, right? To look at all those different markers we were talking about: inflammation, hormones, chronic infections, cardiometabolic panels, genetic testings looking at the ApoE4 gene, right? Because that can kind of change the way you go about things. Cognitive testing, and then also different types of neuroimaging testing, right? We'll talk about those a little bit, but not too much. There's some cool neuroimaging techniques, especially the neuroquant, which is takes an MRI and then it does uses what's called volumetrics, and it measures the volume of all these different regions of the brain to see where do you fit in a percentile region. So specifically, the hippocampus or the temporal lobe is affected in Alzheimer's, so it will measure yours and say, where do you fit in? Are you in the 99th percentile? Are you in the 1 percentile? A lot of the patients I see with dementia and Alzheimer's, they're in the 1 to 10 percentile, right? The volume is shrinking. And so if we can catch those things earlier, we can say what's going on. And then we can also measure progress, right? Which I think is really, really cool. And then things like PET scan, or there's a company called Sarascan that uses PET scans as well, or SPECT scans to look at glucose activity within the brain, right? So this is your cognoscopy, okay? So like we kind of summarize, right? There's multiple different types. There's inflammatory dementias. There's glycation, right? Or blood sugar components. Trophic, so hormones can affect it, right? Miracle grow of the brain. And then you have infections and toxin exposure, right? And we didn't talk too much about exercise, but in a second we'll talk a little bit more about it, about kind of some of the benefits that exercise can play a role in some of these signaling molecules as well. Okay. So your plan. So what's the plan, right? We talked about a ton of things. And like I said, I know we blew past a ton of information that we could spend an entire weekend and a whole week on. But I want to kind of give you a summary of what is your plan? What are things you should ask your doctor for? Or here are some of the things that if you were to work with us or if you were to come see me, right, we would go through and we would test and look at, right? So the first thing, right, is get tested. Get these things checked out, right? Insurance a lot of times will cover a lot of these things. Other times, right, you have to, you pay out of pocket, right? But don't have to spend terribly too much to look at a lot of these things, and they can tell you a ton. And when it comes to brain health, you look at, Alzheimer's patients. My grandfather had Alzheimer's, and he cost my family upwards of $1 million. Not my family directly, but his children. Over the 10 years he had it, he turned multiple families bankrupt. They lost both of their houses, right? So spending, a few $1,000s up front in the early stages, right, to save you millions in the long run, I'd say are a worthwhile investment, right? So we're looking at things like homocysteine, CRP, what's called a cytokine panel, which looks at interleukin 6, TNF alpha. So these are inflammatory molecules secreted by the immune system. So you can see them with chronic infections. Looking at the trophic factors. So looking at a full thyroid panel that includes free hormones and antibodies. So a lot of people will just run what's called TSH. Very incomplete picture of the thyroid, right? You need to be looking at, upwards of 7 different markers on a thyroid panel to really get a full picture of what's going on with your thyroid. And then looking at things like estrogen, testosterone, free testosterone, progesterone, all these different hormones to see, are these things optimal? And if not, trying to make them as optimal as possible to push you towards that synaptogenesis model versus synaptoclastic activity of breaking down those brain cells, right? And then vitamin D, B vitamins, okay? And then when we look at that glycolytic model, right, we're looking at things like fasted glucose, fasting insulin, full lipid panel, and then also an advanced lipid profile looking at oxidized LDLs, LDL particle size, plaque markers, CRP, right? Really taking a picture into more of that glycolytic model, right? So you see where we start getting a lot of this testing, we can start fitting individuals and say, hey, you fit more into a glycotoxic model, or this seems more toxic, or hey, you got a little bit of everything. All 36 holes need to be filled, right? And then we have the toxic. So now we're looking at tick-borne illness testing, viral infections, mold testing. There's a few companies like Cyrex that have a really cool panel that looks at more of that autoimmune model of Alzheimer's, and then another called the Neural Zoomer, which is out of another company called Vibrant, which looks at it from a similar model, right? So there's a lot of cool lab testing, and there's so much cool stuff out there to really get a big picture of what's going on, right? Because the more data you can get, the more changes you can make, and the more you can increase your odds, right, of beating things like this, right, or improving symptoms versus, a lot of individuals are looking at 1 or 2 markers, and those really aren't going to do much. Okay. Also continue with your plan, right? Getting tested, right? Cognitive testing. So you have things called the MOCA or the Montreal Cognitive Assessment. You have things like CNS vitals and also neuropsych testing, right? So it's not bad to find a practitioner. And once again, we use a lot of this same testing to look at which one of those domains are affected, right? Because one big difference between us and other clinics is that not only do we work on the metabolic aspect, we also play a role in trying to activate and rehab those different domains. So working on visual spatial memory, working on all these different things, right? So really kind of making that whole approach, right? And then the imaging that we talked about, neuroquant, and then PET, and then SPECT scan as well. And then genetic testing, right? Looking at that ApoE4 gene, right? Because that gives us a picture of what model do they fit into, right? The better we can classify and the better we can see really what's going on with your physiology, the better treatment plan can be created, right? And the better outcomes will occur versus just read a lot of individuals say, oh, take fish oil, take resveratrol, take curcumin, maybe, and those things are great, but we have to understand why those work and what people would benefit most from those, right? So I'm arguing that we need a more comprehensive, specific plan, right? And that's why I'm not going to be able to give you the recipe for success for treating Alzheimer's, right? Because it's so patient specific, right? We really have to figure out what's going on and then create a tailored plan for that patient, right? But some things that you can have in your toolbox that are kind of things that have been pretty well studied and are relatively safe and can have profound effects, right? And one thing is going to be fasting, right? And this, in a second, I'm going to talk about is going to be my next webinar we're going to talk about, right? But there's different types of fasting, right? There's intermittent fasting, there's prolonged fasting, there's extended fasting, there's calorie restriction, there's things like fasting mimicking diet, right? Right. And you can see all these have to do with going certain periods of time without foods, right? Intermittent fasting, right? Usually eating within a window of 8 to 6 hours, right? So you're fasting anywhere between 12 to 16 hours a day, right? Or fasting up to 24 to 48 hours, right? And I'm not going to get into a ton of the nitty gritty information because that's what the next webinar will be about, right? But fasting is so powerful in changing a lot of these cell signaling pathways. So remember we talked about the pathways that tell us, is this a good environment? Do we want to survive? Is this a bad environment? Do we want to downregulate? Fasting is such a powerful tool in manipulating and tweaking those in a beneficial way, right? So this is something really, really easy. I'd suggest for most people, of course, you always want to check with your doctor before partaking in any of these things. And some of these you need to be under a doctor's supervision, but maybe ask. See if they'd be good for you. And like I said, join me for the next webinar. We'll talk a little bit more, but these are very, very powerful tools. Okay. Oh, got to go back one more. And then also diet, right? And like I said, we could do a whole nother seminar, a week-long thing on different types of diet, ketogenic diet, paleo diet. For the sake of this talk in dementias, right? There's a lot of good research on the ketogenic diet, right, which goes along with the fasting. But I like things like the paleo and the autoimmune paleo diet, right, because they cut out a lot of inflammatory foods. So going back to it, anything that kicks up inflammation, right, has a chance of moving you towards more of that synaptic breakdown versus synaptic growth, right? And this is why we like a lot of fruits and vegetables, and then, right, the meats we do eat, right, highest quality meats, right, grass-fed, pasture-raised, right, really, really good fats, things like coconut oil, avocado oil, olive oil, things like that are really beneficial in rebuilding and building up synaptic genesis, right? Or building the synaptic pathways, right? So like I said, we're not going to get crazy in depth with those, right? And then like we talked about exercise, right? There's so much great research looking at exercise and increase in BDNF. The big way exercise really helps the brain is that BDNF, or that brain-derived neurotrophic factor. So exercise helps produce a ton of miracle grow for your brain, right? So it helps you create more of that synaptogenesis, right? Which is really, really, really cool. But the key, honestly, with all this is to work with the physician to analyze his lab testing to make a specific treatment plan for you. Like I said, with patients, we look at all these lab testing with a lot of neurological testing to really say what areas seem to be affected, what biomarkers are affected in their blood work, and then we do everything to try to optimize those metabolic pathways. But then we also do a lot of things to rehab those different areas of the brain, right? And they're all tailored for that patient. So like I said, there's a lot of things that you can do, right, without a doctor, right? But I highly suggest finding a trained physician to work with that can work on these things, okay? And so the next seminar we're going to be talking about is going to be on September 17th at 6 p.m., right? And we're going to talk about the science of fasting, right? So we're going to kind of continue around the theme we've been discussing with cognitive decline, and we're going to start teaching you the tools that we talked about in this seminar, right? And we'll have more things that kind of build along the dementia, right? So we won't end here. So I'll start going the more in depth with the tools that we briefly discussed here, right? And the big thing that I want to kind of offer you guys is, right, talking through this, a lot of this can be really difficult to understand, or how do I find a person near me, right? So what I'm kind of offering everyone who's listening to this webinar, right, is the opportunity to sign up for a free consultation, right? It can either be in person or over the phone. So this is an opportunity where you and I can sit down, or myself and a family member can sit down for 30 minutes, right? We can go free of cost. We can go through all your lab work. I can help kind of guide you, explain what's going on. I can suggest other testing that needs to be done, right? And we can kind of talk about what is an appropriate plan, right? So this is a really, really great opportunity to sit down and learn a little bit more, ask some more questions. Like I said, this is an amazing opportunity for this free consultation. These go by really quickly. We book up really, really quickly. So what I'm going to do is I am going to post in the chat a link, right? So here is the link. If you guys click on that link that is in the chat, that will take you straight to my calendar, where you can go ahead and you can schedule that and sign up for either in person or over the phone. Like I said, free of charge, nothing. All it costs is your time and my time, and we can chat, right? And you're not going to be pushed into anything. It's just a time for us to get to know each other more and for me to answer more questions for you and see if hopefully I can be of help to you or a family member. Because like I said, this really affects individuals, and I think we're in a really cool time where a lot can be done. So I really suggest if you're curious, reach out. You can also give our clinic a call. It's 303-781-5617. And Lindsay can call you to get something set up as well, or set up a time to chat with me. And then you have my email here, drmaynard@integratedhealthdenver.com, where you can contact me as well. But I want to thank you guys all for joining. I'm going to kind of look through some of the questions real quick. But if, like I said, if some of you guys are interested, I'd highly suggest clicking on that calendar to set up a consult, because like I said, those are going to go by relatively quickly. We fill out pretty quickly. So thank you guys. And I'm going to kind of sift through some of your questions. So Kevin, um, cool. That's awesome. I see you came to the intermittent fasting, that it seems to help. Yeah, I love it myself. I find it really beneficial. I work in a lot of 24-hour fasting myself, intermittent fasting. Once a quarter, I do a 3 to 5 day fast as well, kind of as a reset. So that's awesome. I'm glad that that's definitely been helping. John, have you heard or used BrainSpan? I have not heard of BrainSpan before, but I'll probably look it up after this and see. It might be something I've heard of or used, but maybe it's under a different name. But I have not heard of BrainSpan before. Um, let's see if there's any other questions. Bum, bum, bum. So what about brain spotting? Sarah asks, right? So brain spotting, or some of the brain spotting I'm familiar with, is more in regards to, and you can correct me if I'm wrong, but this is some of what I'm kind of aware of, is brain spotting in regards to like neurofeedback or QEEG. So I know some individuals will use QEEG, right, to look at brainwave activity. So I think QEEG is really, really cool. I think there's a lot of people doing it in a lot of different ways, some better than others, some not really so great, right? So I think the best is like a 19 channel QEEG is what they use in research. And what that does is it looks at brain waves, right? And it looks at how well the brain is oscillating, right? So think of the brain, think of networks, right? And all these networks oscillate at different frequencies. They shut down, they turn on, they go on, they go off, and people can get stuck in these different oscillations. And that's where things like neural feedback can kind of shift those oscillations. Now, that being said, I think that's just one piece of the puzzle, right? If you weren't to affect these other things that are creating beta amyloid plaque, right, it's really not going to be that effective. And then I see the EMDR. So I'm familiar with EMDR, especially when it comes to traumas. I think things like EMDR is really effective for working through traumatic events and things like that. So I really like that, and we use that a lot in our clinic, or our therapists use that a lot to help patients work through traumatic events. And the diagnosis of Alzheimer's or degenerative disease is very traumatic, and that stress signal is another thing that's going to create more of that beta amyloid plaque. So I think that is important to look at. Let's see, Kevin. I'm glad, Kevin. And I think you're totally, totally right, that the earlier you can do things, the better. So glad that that's been helpful for you, and I'm sorry for the things with your family as well. So cannabinoids. So yeah, so I think that CBD has some promising things. I'm not necessarily crazy about THC when it comes to dementias, right? Because of how it affects, especially the hippocampus, right? So the hippocampus is the main area affected in Alzheimer's, right? That's where short-term memory, visual spatial memory occurs, right? And there's some stuff looking at, right? THC affects those areas, right? And it downregulates, it changes oscillations in that hippocampus. So I'm not crazy about it in that regards, but CBD, I think there's some cool research coming out because CBD seems to have more of a immune modulatory effect, right, and an anti-inflammatory effect. So I haven't seen anything in regards to how it may affect that SNP in that peptide in the creation of beta amyloid plaque, but we definitely use CBD a lot in a lot of different cases, and I think it could be powerful, and we'll learn more definitely. So, any other questions? Let me see if there is another. Oh, sorry, Dana. I see you asked some questions as well. Um, so does brain injury, can that cause this illness? So, there's a lot of debate and discussion around that. So when you look at traumatic brain injuries, right, traumatic brain injuries can definitely set the stage for neurodegeneration, right? Things like Parkinson's, things like Alzheimer's, right? Can we say that that's the exact cause? No, right? But a brain injury is going to shift you towards more of that synaptic clastic activity. It's going to create a lot of inflammation, a lot of immune regulation. Now, that being said, right, like I said, we work with a lot of brain injuries that it doesn't have to end up that way, right? If a brain injury is treated appropriately and an individual heals from that appropriately, it doesn't have to end up in something like Alzheimer's. But multiple brain injuries can definitely increase the risk, right? Because it's just creating destruction in the brain and inflammation as well. Okay. So, what was the first test to realize that something is wrong in the brain? So, um, I'll try to my best to answer this, right? So, there can be, like I said, we talked about a lot of lab testing. I think one of the biggest things is, right, more so probably the subjective cognitive impairment. So, when individuals really start noticing, saying, things just don't feel right. My memory's not quite where it should be. I'm forgetting things, right? Those early things that a lot of people write off and think, ah, that's just old age or this, that. Those are early signs that should be paid attention to, right? Those aren't normal and they shouldn't be normal, right? They may happen once in a blue moon and it doesn't mean it will end up in Alzheimer's, but it's a sign that things aren't optimal in the brain, right? And we should start digging to say, why aren't things optimal, right? Because you shouldn't be walking into a room forgetting why you walked in there, right? So those are some of the early signs that can kind of give us an idea that something's going wrong in the brain. So I hope that answers your question, Diana. And if it doesn't, maybe re-ask it and I can see if I can answer it in a little bit better of a manner. Um, let's see. Any other questions, guys? So good question. This kind of comes back to the some of the subtypes we talked about. So if you look at some of the research, especially Dale Bredesen's work, he'll discuss that the different subtypes present at different stages of life. So type 1, inflammatory, that has more of a genetic predisposition, tends to happen a little bit earlier. These are the individuals where it occurs sometimes in the late 40s, early 50s, late 50s, so early on versus some of the type 2 or the non-genetic types tend to happen in later decades. So early 70s, early 80s, so a little bit later on. And then you have the type 3 or the toxic, and those are kind of like the genetic forms where they can start a lot earlier, right? So once again, in that 40 range, in that 50 range. So whenever you see an individual where, 40s and 50s and things don't look so great, I'd be quickly asking myself, is there a genetic predisposition? What's going on with their genes? Is there family history? Or more of that toxic model, right? Because that's too early a lot of times for a lot of these metabolic pathways to break down, right? Because remember, the biggest the biggest thing that increases your risk of dementia, right, is time. It is aging, right? So I'd say that's where I'd start looking at those toxic models. So from a standpoint of testing, the big things I would look at are different types of infectious disease. And once again, a history is key, but this is where you're looking at things like Lyme disease, different forms of tick-borne illnesses like Babesia, Bartonella. You'd look at mold or mycotoxins. You'd look at heavy metals and chemicals, right? So there's different labs. So IGeneX is a great lab for tick-borne illnesses and Lyme disease. So is Vibrant America. Um, there's a lab called Great Plains Lab or GPL, who has a really great tox panel. So looking at different toxic chemicals. They also have a great mycotoxin panel that looks at urine metabolites of mold. Vibrant also has that same panel. And then a company called Cyrex has a great panel that looks at are you making antibodies against your brain that could be creating kind of like a toxic autoimmune Alzheimer's type presentation? And Vibrant has those as well. So those are some of the things to look at are infections. So viral infections, bacterial infections. Also another thing, certain types of dental infections. Actually, there's a lot of research looking at different types of dental infections and periodontal disease actually being correlated in increased risk of Alzheimer's. So some of the bacteria associated with that as well. If that helps answer your question. Um, so kind of field a few more questions, and if no one else has any more questions, then I think we'll kind of end it there. Like I said, there will be a link if people want to rewatch this, and you should have access to my presentation as well, so you can kind of go through all those different testings we had. Like I said, if you want to learn more, feel free to either call the office, click on the link, and set up a time to speak with me. Otherwise, thank you guys so much for joining. I hope that was helpful. I hope that this helps either you guys or a family member kind of get help sooner than later. Because, like I said, this is a real epidemic, and I don't think it needs to end the way it's ending. I think a lot can be done. So one more question. So by Allison. So with fasting, does one need to be stabilized beforehand? Are you meaning in regards to like blood sugar, like stabilizing blood sugar beforehand? Because if that's the case, then yes, I would say, stabilizing blood sugar and things like adrenals and hormones is definitely priority before working into fasting. Granted, fasting can be a method to stabilize those things as well. So it just is patient specific, but if you do have blood sugar issues, hormone issues, adrenal issues, you do want to take caution when doing fasting because fasting can make hormone issues worse. That being said, it can make them better. So if you do have issues with blood sugar, adrenal hormones, I definitely would speak to your physician or person you're working with before partaking in it. So, yep. Let's see, we got another question from Diana. So how can someone that eat every 3 hours fast? So great question. So it depends whether that person eats every 3 hours because they think it's good for them, or they eat every 3 hours because they can't last without it. Right. So I always take it as a sign that if an individual can't go more than 2 to 3 hours without eating, that's a problem. And that although that can help them in the short term stabilize their blood sugar, that it's just covering up a symptom, right? So the fact that I think most individuals, if healthy, right, they should be able to go 24 hours without eating with little to no side effects. Now, once again, I think that's optimal. And individuals who can't, right, it's because there's issues in metabolic activity and energy demands, right? So their body's not efficient at using energy reserves, things like that. So one kind of strategy that I'll use sometimes, right, is you have to look at the foods individuals are taking, right? So starting to move towards more low glycemic foods or foods that don't really spike blood sugar, adding more fats and proteins into meals to stabilize blood sugar. But how I kind of start with individuals is I start having them eat all of their calories kind of in a 12-hour fasting window, right? And then the more stable they become, the slower we start to narrow that window, right? So we're slowly building metabolic stability, but also starting to create that endurance and that fasting. So there's a lot that can be done to stabilize blood sugar and to move someone into fasting. But I think if you have to eat every 3 hours, otherwise you get symptoms, then eating every 3 hours is not, I wouldn't say the solution. It's kind of what's helping keep you stable, but you should be able to go without food. So it means there's things aren't fully fixed or healed yet. And I think you should be worked towards that by whoever your doctor is or whoever is working with you. And like I said, that's something we'll talk a lot more in detail during the fasting seminar. Okay. All right. Any more questions? If there's no more questions, then I think we will end it there. So once again, thank you all so very much. I look forward to hoping to see all of you guys in the next month at the next webinar. We're going to try to do more and more of these. And please, if there's topics you guys want to learn about or hear about, please let us know. You can email me and we can see if we can put together a webinar for that as well. Perfect. So, like I said, in regards to some of the testing, there should, like I said, you guys should have access to this. So you have some of the things in regards to links. What we may be able to do, what may be great is if you're interested more in testing and some of those things, right? You can always kind of call the office and you can talk with Ruth, and they can always kind of talk you through those different labs and things like that as well. So thank you guys so much, and I hope all of you have a great evening.